Okay, hi everybody, this is Lilith again. I'm one of your IAs for BIPN 160, and this is part 2 of lecture 10. Okay, part 2 of this lecture is more research heavy, so please hang in there with me while I try to do my best to explain the main points of each research paper. And there will be some parts where I will just ask you to memorize it, but I promise that it will be for your own good, right? There will be just easy things that you want to memorize that might serve you well for the exam. So let's get started. The second part, or at least where I stopped in my first part of the video, I stopped right before the dorsal raphe and its production of serotonin. So let's continue from there. So dorsal raphe is a nucleus that is located right over here in the pons, and it has, through this picture, many different projections. But the most important function of dorsal raphe that we want to remember is that it releases serotonin, and that serotonin is released precisely when reward is given. Right, so we will talk more about this in a couple minutes, but if you remember back, dopamine was also involved in reward signaling. However, dopamine was involved in more reward prediction, whereas serotonin here isn't really released during reward prediction, but it is released right when the reward itself is given. Okay. Some other key moments from the lecture about serotonin is that the neurons, the the neurons that produce serotonin show preference behavior, meaning they show liking behavior. And I didn't include the picture from the research study here, but it's right after this picture in the lecture notes. So if you're looking at it, it is the mouse where they are activating the neurons that produce serotonin, and they only activate those when the mouse is located in the box in the center of its field. So when those neurons are being activated, then the mouse loves to just hang around in the middle because it likes to have the feeling of serotonin being produced. And because the mouse chooses or prefers to go to the middle of its playing field, then this confirms the fact that activation of serotonin neurons show preference behavior in the animal. Okay, and then another key point, or I'm not even sure if this is a key point. I think Dr. Root just mentioned it because it was interesting, but might as well. These neurons not only produce serotonin, but also produce glutamate. And so there's still a lot of research going on in the field of what the difference between glutamate and serotonin stimulation is. But I think for this class, for the scope of this class, just knowing that glutamate and serotonin could possibly be working together to be producing these types of feelings in the mice that are tested, and that should be enough. So there isn't one or the other, they're more of a team together, and they work together in this feeling of reward and pleasure. Okay. And then Dr. Root put together 3 key points that are comparing dopamine effects and serotonin effects. So again, going back to the fact that dopamine is more for prediction of reward, and then serotonin is for reward itself, reward or pleasure. So I'll go ahead and read this. Dopamine neurons are firing in a very transient way, and in a predictive way. So they fire when the animal is expecting a reward, whereas serotonin neurons fire much more closely to the reward. Okay. Number 2 comes from an experiment that was done. So sucrose and cocaine can produce liking behaviors when dopamine is blocked. So even when dopamine is blocked, then liking behaviors are still being produced. So this tells us that we don't even need to have dopamine to have that liking behavior towards molecules such as sucrose and cocaine. So that tells us that serotonin actually has a big role in this liking behavior, and it's not just dopamine. And number 3, disruptions of serotonin causes depression and anhedonia, whereas dopamine causes disruptions in motivation. So depression and anhedonia, I think it was lack of feeling pleasure, and depression, you can think of it as the opposite of feeling rewarded. Okay, so when we disrupt production of serotonin, so serotonin's gone, we feel the exact opposite of reward and pleasure. And when dopamine is gone, we feel the exact opposite of being motivated or being or expecting some kind of reward. So again, number 3 goes ahead and solidifies the main idea that dopamine is reward expectation or prediction, and serotonin is released right when the reward is given. So I'm really trying to hit home with that message. I think personally it will be at least one question when you're tested. Okay, and a summarizing point, dopamine seems to be more related to reward prediction and motivation, whereas serotonin is involved in reward and pleasure. Okay, so I think hopefully I drove that home enough. Then we started talking about the amygdala, and the amygdala is located on the inner temporal side, right next to the hippocampus. It is involved in fear, emotions, liking or disliking behavior, which is another way of saying valence behavior, and it is also involved in learned behavior. Okay, so that's kind of an overview. Before we move on, I've put together the inputs and the outputs of the amygdala. A student previously asked me last week if you all should be memorizing the information in the boxes or the information that Dr. Root listed over here on the side. And at the time, I told the student that you should look at both, and to some extent that is still true. You should read all of this on your own, but when I watched the lecture more carefully, it seemed like Dr. Root was mentioning most of these more rather than reading what is in the blue boxes. Okay, so I would say read the blue boxes, but definitely memorize these. Okay, and before I move on, this yellow blob depicts the whole amygdala, the organ or the brain area of amygdala. And inside amygdala, we have many different parts of amygdala. And some of the parts that will be important in the next coming topics are the basolateral amygdala. And basolateral is the basal and then the lateral. Sometimes I guess maybe this whole area is called basolateral, I'm not sure, but just remember BLA, basolateral amygdala, and also central amygdala. Central amygdala is written CEA. Okay, and we will get to talking about those more. One more important thing from lecture, if you remember, Dr. Root asked about 2 areas in the amygdala, and I'll go ahead and highlight them. I think one of the questions was about the lateral amygdala and how it receives a lot of sensory information from the thalamus and other sensory areas in the brain. And then another key output that he talked about was central amygdala, and how central amygdala has a lot of outputs in this list, he only stated hypothalamus and brainstem, but here in blue you can see that the central amygdala actually inputs to a lot of different places. So I would just remember sensory information goes into lateral amygdala, and a lot of output comes out of central amygdala. Of course, you should pay attention to the other ones also. Okay. This study shows that amygdala is actually also involved in displaying learned behavior, or I don't remember if this was learned fear to be exact, but it was learned behavior. So this left side of the graph shows that when the researchers would train the mouse, they would train, and then the mouse would learn a behavior, and then if they stopped training, then extinction would happen. And extinction of a behavior is just when, if you don't train the mouse for a long time, then the mouse just forgets how to do a certain behavior. Okay, so this is normal, just having normal amygdala. You train an animal, and then after some time, the behavior declines because you stop training. And then right at this dotted line, what they did is they lesioned the amygdala, or a part of the amygdala. And what they noticed is that even though they lesioned the amygdala, there really was no change in the actual learning ability of the mouse. See how in the beginning they still learned, and then after we got extinction. Okay, so the actual learning ability, or ability to learn, did not get hindered by lesioning the amygdala. However, what did change was the ability to display the learned behavior. And what does that mean, to display? Meaning if you teach a mouse to do something and then you lesion their amygdala and then have them do it again, they won't be able to. And that's depicted in part B of this study, on this right side. So here again, they taught the mouse something, so the behavior is going up. Oh, in this case it's freezing, so it is learned fear. Yeah, when the mouse is fearing something, when it's afraid of something, then they're freezing, right? They just stand there being afraid. So in the experiment, they trained the mouse to be afraid, and now it's going well. And then they took some of the mice and they lesioned a part of their amygdala. And then when they played the sound again, they saw that the mouse was no longer afraid. See, the percent of freezing has declined, meaning after the lesion of the amygdala, the mouse no longer remembered that, hey, I was trained to be scared of this sound. Okay? So that's what the black dots show. The white or unfilled dots show a sham experiment. A sham experiment is when you take some of the mice, and instead of doing surgery on them and lesioning their amygdala, you do some other surgery that doesn't lesion their amygdala, and it's a scam basically, right? So they still did surgery on them to make sure that the surgery wasn't the important factor in the results of the experiment, but they didn't actually lesion their amygdala. And you see over here that the mice that went through the sham experiment, the sham surgery, were still able to show some kind of freezing until extinction of that behavior happened. So hopefully that made sense, but I've summarized it here for you if you need to listen to it in a more compact way. So amygdala is involved in learned fear. However, when the amygdala or a part of it is lesioned, the animal cannot display its learned fear anymore. But a lesioned amygdala does not hinder the animal's ability to learn. I'm missing an apostrophe there. The animal's ability to learn. Okay? And then I didn't include the monkey experiment. If you're looking at the lecture notes, they used a lot of monkeys and they just put boxes full of bananas and they were excited when they would see the banana or something. But then sometimes they would put a rubber snake and they would just get scared of the snake. However, when they lesioned the amygdala of the monkey, then they had less fear whenever they would open the box and they would see the rubber snake. So again, same results of lesioning are shown in the monkey experiments. Okay, hopefully that's a good compact summary of the main points. Now, so far we talked about the amygdala. We talked about a couple things. We talked about how it is involved in fear, how it is involved in learned behavior, but we haven't really talked about emotion or valence just yet, right? We haven't talked about liking or disliking or feeling any positive or negative cues. And this is where these studies help us understand how the amygdala responds to positive and negative cues, such as responding to a molecule of nicotine, which makes them feel good, or a foot shock, which makes them not feel good, right? Be scared or just not a good feeling. And just like if you think back to the part 1 of lecture 10 of my review video, we talked about how the nucleus accumbens had distinct neurons that responded to good things and bad things, right? The VP, the ventral pallidum, had distinct neurons. So just like those brain areas, the amygdala also contains distinct neurons that respond to either a good stimulus or a positive stimulus, and then a negative stimulus. So let's go over this experiment. I think I understand it. I'm not sure if to be honest, I didn't get this experiment too well when Dr. Root explained it, so I had to watch it a couple times, but I think I understand the gist of it, so let's go through it. What helped me understand this study best is realizing what these side legends are, what the side pictures are. So the yellow shock, it stands for the foot shock. So that's like something preliminary that you should understand. And then the needle is showing a nicotine injection. Okay, and remember that the mice like nicotine, so it's a positive cue, and then they don't like foot shock, so it's a negative cue. Also pay attention to the times that these shocks and shots of nicotine are given. For example, this says right here 0 hour, and then this says 18 hour. So this happens first, then this. Same here, this happens first, then this. Okay, so that's some preliminary things. And then another thing that you should think of or review is how CFOS works. So if you remember, CFOS is a gene that is activated in neurons that are working, or in neurons that are being activated. So if I activate a certain neuron that has the gene CFOS in it, then that neuron will have its CFOS genes activated. In this case, the CFOS is connected to channel rhodopsin with a YFG, which is portrayed as green. So when this is activated due to neuronal activation, then the channel rhodopsin that's attached to YFP will also be activated, will also be expressed, so we will see the green stuff. Okay, and then M cherry will also be expressed, and M cherry is just another marker that shows up as a red when it is expressed. Okay, so after that review, hopefully I can explain this well enough. So what happens in this experiment is the researchers first take a mouse and they give the mouse a foot shock. Okay, they give the mouse a foot shock, and if you think about it, a foot shock is a stimulus, right? And if our hypothesis of the amygdala containing distinct neurons is correct, then when the foot shock is given, a certain type of neuron that is responsible for responding to a negative cue, such as a foot shock, will be activated, right? And only those quote-unquote foot shock neurons will be activated. Okay, so when the foot shock is given at 0 hour, then we get production of M cherry, for example, over here. And we see that, hey, these neurons are red. So these neurons are responding to a foot shock. Okay. Then, I think what Dr. Root said is that this green stuff, I'm just going to call it green stuff because let's just simplify it. The green stuff is also being produced, but after some time, it fades away. Only the M cherry stays. Okay, so after some time, after 18 hours, the green stuff fades away. However, 18 hours later, they give the foot shock once again, and after they give the foot shock, they actually go and stain the CFOS, and I want to say this YFP also. Just know that they stain it, so they are able to visualize it. Okay, now this is no longer faded. Now we can visualize it. And when we visualize it, we see that it is exactly the same neurons being activated for the second foot shock as it was for the first foot shock. Okay, so there is 100% overlap between the neurons that are activated in the first foot shock and the neurons that are activated in the second foot shock. Okay, and that is a good sign, meaning that whenever you give the mouse a foot shock, it is always going to be the same neuron that is activated. So that solidifies the theory that there indeed is a distinct neuron that responds to foot shocks or negative things that happen to the mouse. Okay, and then they did the same thing with nicotine. They injected nicotine at hour 0, and they saw that these neurons are labeled. So then they said, cool, let's go sleep. We'll come back the next day. 18 hours later, they give the mouse another shot of nicotine. And then they stain for CFOS, and they see that, hey, the same exact neurons that were activated yesterday by the first shot of nicotine are activated here by the second shot of nicotine. So again, this confirms the idea that there is a distinct neuron that will get activated every single time you give the mouse nicotine. Okay. Now, they did the third phase of this experiment. And during the third phase, they first shocked the mouse with a foot shock at hour 0. And at hour 0, they visualized using M cherry that these specific neurons responded to the foot shock. So then they said, cool, let's go sleep, come back the next day, 18 hours later, now let's give them a nicotine shot. And when they gave the mouse a nicotine shot, they saw that these were completely different neurons that are marked green now, that are activated by the nicotine shot. So this was absolutely zero overlap between the neurons that are activated by the foot shock and the neurons that are activated by nicotine. So this tells us that there are two completely distinct neurons, one responding to shock or foot shock, another one responding to nicotine. And then in part 4 over here, same concept, just changed stimuli. So first they gave the nicotine, they saw that this neuron responds to the nicotine. 18 hours later, they gave the shock, and then they said, oh, different one. So this one that I'm highlighting and this one that I'm highlighting on the left, they're not the same neuron, meaning that nicotine and foot shock are being responded to by different types of neurons in the amygdala. Okay, hopefully that made sense. Main takeaway, these are different neurons. Okay. Then I think these are kind of, oh no, never mind, not random. So if you remember the area BLA in the amygdala, basolateral amygdala, this research study showed that BLA is involved in learning, okay? And how exactly did they test this experiment right here? What they did is, I think I'm going to give you another summary that is mainly depicted over here because I don't think Dr. Root talked about it really. But to set up the experiment in your head, what they did is they allowed the mouse to, I think, be in a little box, and then on the left side, or on one side, you have one type of smell, just a random odor, and then on the other side, you have another type of odor. And then if the mouse decided to go to, let's say, odor number 1, the researchers activated a specific neuron. So they either activated the foot shock neuron or the nicotine neuron. And depending on which neuron they activated, the mouse began to either like that odor or dislike that odor, right? And the important part here is that the odor doesn't matter. The mouse would go in there. Let's say it smelled like roses. If the mouse went in there and it smelled the roses, it would originally like the roses. But if the researcher decided to activate the foot shock neuron in the mouse's brain right when the mouse was exploring that odor, the mouse would avoid that odor at all costs because it is getting the stimulation in its brain of that foot shock neuron, right? It's getting negative stimulation. So it automatically thinks that the odor that it's smelling is bad. So that's what kind of this is showing. So approach, avoid in text, I think when it's a negative number, then it is avoidance. And then when it is a positive number, it's approach, meaning it likes the odor that it's smelling. And see, color-coded, red is a shock, a foot shock. So when a foot shock was given, the mouse would think that this odor is a really bad smelling odor and it would dislike it. But when the nicotine neurons were activated, then the mouse would actually like the odor and approach it more. And I've summarized it here in one sentence. Activation of specific neurons will let the mouse have preferences over random odors. And random as in the mouse not necessarily responds to the odor itself, it just responds to which brain area or which neuron in its amygdala is being activated. Okay. Main points on BLA, basolateral amygdala, from Dr. Root's lecture. This would be something good for you to memorize. Definitely recommend this. BLA contains distinct neurons that are activated by positive and negative stimuli. Exactly what we talked about right here, right in this experiment. Distinct neurons. These neurons are sufficient to produce behaviors consistent with liking and disliking. This is also what we talked about, right? A foot shock will display disliking behaviors, and then the nicotine will allow the mouse to feel that it is liking that stimulation. And then these neurons in the BLA can also be used to assign valence to sensory stimuli. So this last sentence has to do with this experiment. Using these neurons, we can make the mice learn to either like a sensory stimulus, for example, a smell, an odor, or to not like it, depending again on which neuron we activate, the foot shock neuron or the nicotine neuron. Okay, and when we say foot shock neuron or nicotine neuron, again, these aren't the names of those neurons, right? It's just nicotine is a molecule that activates the neurons that make you feel positive things, and then foot shock makes you feel negative things. Okay. And then there are a few I felt kind of out of place research studies, so they don't really go with the flow of what we just talked about, but let's go over it anyways. This is the part where I will ask you to memorize things because Dr. Root didn't really explain the techniques of these studies, so that's okay. This is Dr. Tai's research. If you're following with the lecture notes, then you can find which one that is, the title and everything. And Dr. Root just talked about the result because this is still an ongoing research project, so we didn't really go over the techniques. But you definitely want to memorize this. I remember this was on my either exam, or I remember seeing this type of question somewhere. So just remember that the basolateral amygdala, we just talked that it has neurons that respond to positive cues and neurons that respond to negative cues, right? So where do these positive and negative cues project? Well, from the BLA, the positive cues project all the way to the nucleus accumbens. So the nucleus accumbens receives input from BLA, and this is strictly reward input. And then the central amygdala receives input from the BLA strictly on aversive cues. So negative stimuli. Okay? So plain and simple, BLA projects to nucleus accumbens. Good, because reward. BLA projects to central amygdala. Bad, because aversive or negative valence. Okay. Then we went on to talk about the projections from the taste cortex or gustatory cortex, projections from taste cortex into the amygdala. So let's go over this. If you remember from a few lectures back, the taste cortex actually has specific sweet and bitter cortices in that area. Okay, so if there is a sweet cortex and a bitter cortex, then there are also sweet and bitter projections. So the taste cortex has sweet and bitter projections, and each one of those project to different parts of the amygdala. The sweet neurons project to BLA, therefore they allow the mouse to feel liking feelings. And then the bitter neurons project to central amygdala, giving the feeling of disliking. And in this research study, I believe Dr. Root only really talked about this top portion, and let's see what happened here. Licking increased over here. Yeah, so if we stimulated, see light stimulation, if we stimulated the sweet projection from the taste cortex into the amygdala, we see that licks of, I'm guessing just water or some substance increases because the mouse likes how this tastes. However, if the bitter projection from the taste cortex is activated, then you see the blue bars, licks have declined because the mouse no longer likes whatever that substance was that it was previously tasting. Okay, so I would say main takeaway, definitely memorize sweet neurons to BLA, bitter neurons to central amygdala. Easy way of remembering this, or I guess a hint, central amygdala receives negative cues or aversive cues from the BLA, and central amygdala also receives negative cues or feelings of disliking from the bitter cortex. So kind of, you know, when you think of central amygdala, just think not good, right? Disliking, aversive. And then the other ones you just memorize. Another topic that we covered pretty quickly towards the end of the lecture was that if we silence neurons in the amygdala, this stops taste valence. And this was the research study where the mouse was drinking, or the mouse could drink something that is bitter, just plain water, or something that tastes sweet. However, after silencing the neurons in its amygdala, the mouse stopped feeling the difference between which solution tasted bitter and which one tasted sweet. So after silencing the neurons in the amygdala, the mouse feels indifferent about the sweet and bitter juices. Okay? But even though silencing the amygdala stops taste valence, meaning the mouse can't tell if it likes it or doesn't, silencing the amygdala does not stop the animal from recognizing what is sweet and what is bitter. So main point here is that although silencing of the amygdala stops the mouse from understanding what the mouse likes and dislikes, the silencing does not stop the animal from recognizing what is sweet and what is bitter. So the mouse still recognizes which solution is sweet and bitter, but it just feels indifferent about it. It just keeps drinking it, you know? Doesn't have valence towards it. And this point is shown in this experiment. And I've cut out the top portion only if you're looking at the lecture notes, because if I remember correctly, this is the only part that Dr. Root talked about. So in this research study, they allowed the mouse to poke its head in this area, and it would periodically get a sweet tasting substance or a bitter tasting substance, and depending on what it is, the mouse was trained to either go to the left side or the right side. Meaning if it got a sweet tasting molecule or substance, then it would go, for example, to the left, because the left side tasted sweet, to get more sweet stuff. And then if whatever came down this port was bitter, then the mouse would go to the right side to drink something else like water to relieve the bad taste of bitterness. Okay, so they first trained the mouse to go left or right. So they trained the mouse to recognize whether whatever it's tasting is sweet or bitter. And then after training the mouse, they tested the mouse, and they said they realized that it's working great. Most of the time the mouse goes either to the left or to the right when it recognizes whether it's sweet or bitter. And then they went ahead and they inhibited or lesioned the BLA of amygdala, and they saw that there really isn't a difference between the going left or right. So the mouse was still able, even with a lesioned amygdala, the mouse was still able to taste the sweetness or bitterness and then say, oh yeah, I recognize that, that's sweet, so I'm gonna go to the left. Or yeah, that's bitter, I'm gonna go to the right. But it had no valence towards it, meaning it didn't either like or dislike the sweet or the bitter. Okay, hopefully that made sense. Again, main takeaway, silencing neurons in amygdala stops taste valence, so stops liking or disliking. However, the silencing does not stop the animal from recognizing sweet and bitter. Okay, I think that was the last of the research studies and last of the lecture. I know that this lecture was very heavily research oriented, but if I were you, I would just go back and listen to Dr. Root's recording. Hopefully this video is also helpful, but just summarize all of the experiments that he talks about and try to just keep remembering which experiment or which technique pertains to which part of the brain, right? So in this video only, we talked about serotonin, then we talked about the amygdala, right? And in amygdala, we talked about different things. We talked about learned fear, and then we started talking about how the amygdala has distinct neurons, and then we talked about the good type of, the bad type of neurons, the good type of neurons. And then we started talking about using a part of the amygdala, the BLA, for learning again. And then we talked about valence one more time, right? Liking or disliking. Memorize this stuff, memorize this stuff. So just try to summarize all research findings that Dr. Root talked about and just study it by connecting it to the material that we covered. Okay. Here are, I'll zoom in. Here is the practice questions. I'm not going to go over it because the answers are right there. Please email me if you have any questions about them so I can explain them further. But it's just, this video will be too long if I just read it. So you can stop the video here and go over them. You definitely should. And then this is the free response. So stop the video and go over it. Definitely will help you. And then here are the learning objectives, and you should definitely go over these to check yourself if you're missing any type of information that you should know for this lecture. Okay, that's it for this. I'll post lecture 11 and 12 this weekend so that you have it for your exam. Don't forget, the practice set or the problem set 5 is due Friday, and your next test is on the 23rd, I believe, on a Tuesday. Good luck. My name is Lilith. Email me, message me through Canvas. I'll do my best to get back to you as fast as I can. Thank you.